everyone, this is our video about tryptophan metabolism. Tryptophan is an amino acid. It's kind of apropos for Thanksgiving, which is when I'm making this video near to Thanksgiving, because tryptophan is very much found in turkey. It's also found in tofu, which if you're eating a tofurkey is made of tofu, so you're still going to get the tryptophan. And it's also in seaweed, seaweed, but you know, in sushi, there's seaweed. Kelp is, you know, seaweed, and then these lovely looking seaweed snacks. So lots of tryptophan in all of those places. But tryptophan is an essential amino acid, so that means that your body cannot make it on its own. So it cannot transaminate anything in order to make tryptophan. You have to ingest it. Now, once you have your tryptophans, once you've eaten your, let's just say, tofurkey, you will then have it in your body. Your hepatocytes are going to do various things with it, and one of the things that they can do is they can produce vitamin B3, which is niacin. Now, niacin is eventually, with a little bit of changing, it's going to be the precursor of NAD+. We know that we just put a hydrogen onto it to make NADH, and the kind of back and forth between that is how we're shuttling hydrogens in energy creation, or shuttling electrons, I could say. We'll also need that to make NADPH, which we know is part of fatty acid synthesis as a result of the pentose phosphate pathway. So all of this is really, really super important just for energy creation and creating fats. Now, the tryptophan can also be used, depending on which enzymes are present, it can be used instead to make serotonin and then melatonin. So for, you know, neurotransmitters that are handy to have in the brain. Now, what you need to make these things, you have to have tryptophan. You need to have an enzyme called PLP, which is, so this is a little bit misleading. It's not exactly a coenzyme. It's not a coenzyme that works with vitamin B6. It's a form of vitamin B6. So a form of vitamin B6 that is biologically active. So PLP we're going to talk about in a little bit more detail, but PLP is just the active form of vitamin B6. So that's what your body is going to use to do a good amount of transamination, including transaminating your tryptophan to get these things or to get serotonin and melatonin. Now remember that coenzymes, because this would be considered a coenzyme in the reaction in general, coenzymes are important for facilitating various reactions, and in the case of PLP, what it's doing is it's helping deamination. Okay, so here are the two main pathways that tryptophan can take. When we look at tryptophan, here we have two choices essentially. We can go this way, where we're going to make serotonin. This makes you happy. That doesn't look so happy. There you go, happier. Or melatonin, that makes you sleepy. You have no here. Let's give you some arms and legs. There you are, sleeping. Okay, so these are all very calming neurotransmitters. So these are nice to have. These are, you know, things that if you're short on them, you'll know it. If you're short on serotonin, that can lead to depression, anxiety. If you are short on melatonin, that would lead to insomnia and sleeplessness. So this is an important pathway to have. And there are various dehydrogenases that lead to this pathway. Now, for this pathway, the kynurenine, that word trips my tongue, kynurenine. I keep saying it extra, kynurenine. There, Kynurenin. The kynurenin pathway is what leads us to NAD, NADH, NADPH. So this is also important for other reasons, for energy creation and lipid formation. What encourages this pathway instead of this pathway is the presence of a couple of different enzymes. IDO, so there's a couple of IDOs here. That's an important one. KAT, right there, and you can see vitamin B6 or PLP being the cofactor there. But KAT is the main enzyme there. And then TDO, which is right here. So these three enzymes, IDO, KAT, and TDO, are what we call the rate-limiting enzymes. For the kynurenin, God, I hate that word, the kynurenin pathway. So the amount of enzymes, of these three enzymes that you have, will determine how much of the kynurenin pathway you're going to do. Now over here, the dehydrogenases, particularly the tryptophan, or specifically the tryptophan dehydrogenases, these would be the rate-limiting enzymes for this pathway. So again, just back to metabolic channeling, we've seen it a million times. If you have a lot of this enzyme, you're going to do a lot of this. If you don't have much of it, you won't do much of that. And same with over here. So pretty straightforward there. All right, so let's look at PLP. Remember, PLP is the biologically active form of vitamin B6. So you ingest vitamin B6 and your body's going to make PLP out of it, among other things. Now PLP is really essentially an amino acid at heart, right? I should not say an amino acid at heart, it could be an amino acid. The amino acid form of it is PMP. So PMP has the amino acid. PLP is missing the amino acid. So essentially, PLP will steal the amino acid from some other protein, some other amino acid. It will use it, once it steals that amino acid, it will become PMP. Then PMP will donate that amino acid to another molecule, to a keto acid, say, or any other deaminated object, and then we have PLP again because it's just lost the amino acid. So it's just a shuttler of the amino acid. So this takes the amino acid from something, becomes this, then this gives the amino acid to something else, and we have just transaminated. So we've seen this process before where we have the aminated form and the deaminated form of something, and it's just carting that amino acid from one place to the other. We saw it in the malate aspartate cycle with alpha-ketoglutarate and glutamate and aspartate. All those molecules in there were just kind of bouncing their amino acid around. So this is the same exact idea. Now I know you can't see this. This is not super clear. Let me try and zoom in a bit. Still not really good. Okay, so let me just come back out again. I'll just write it. So this is just saying substrate, right? This is just the thing that we're working on, the thing that we're beginning with, or that PLP is going to steal the amino acid from. Let me get a different color. So here's the amino acid. So NH3 is the amino acid. PLP is going to steal that amino acid. And now we have PMP. So PLP just took the amino acid. Now there it has an amino acid right there. So that amino acid just went boop, over there like that. This leaves us with just this keto acid over here. So it's deaminated. Because it's just missing its amino acid. Now, PMP is not going to keep that amino acid. It's going to give it to this keto acid, which is a substrate now. So it's in need of a keto acid, or not a keto acid, it's in need of an amino acid. So here comes PMP, the amino acid goes on to here, and now we have a new protein. This is the product. And it has its amino group. And that leaves PMP. Now that lost its amino acid, so now it's back to PLP. So this can just sit here and do this all day long, right? This is just, transferring the amino acid from one place to the other. Now just keep in mind, we're talking specifically about tryptophan in this video, but this is just generic. This is deamination and transamination of a whole bunch of different kinds of proteins. It could be any kind of protein that we're taking the amino acid off of and just switching it over, putting it onto any kind of keto acid. So, this is a kind of ubiquitous process that's happening all over the place, not just in the tryptophan process. Okay, so here it is again. Same process. There's PLP. There it is without the amino acid. Here's PMP. That is it with the amino acid. So all you're doing is you're taking, again, just follow the amino acid. Here's an amino acid. PLP is stealing the amino acid from this, or stealing the amino group from this amino acid. We're getting PMP, just like we did before, so it's going that way. Right? Leaves us with this little keto acid, lost its amino group, right? Now PMP will donate to our new amino acid over here. So we just transaminated from here to here by adding our amino group to this new keto acid. So this can be any protein that we're making, any amino acid, any keto acid. Now remember, we're transaminating, so we need enzymes for this. It's not just PLP. This is just that biologically active cofactor form of vitamin B6. We still need the enzyme, which is a transaminase, to facilitate PLP's ability to steal this amino group from the amino acids. Okay. Okay, so now how do we take this and take our tryptophan? So there's a lot of words in here and they're very small, and I know you can't really see them very well, but the whole point of this slide is how do we get from tryptophan all the way down to NAD plus, and then finally adding a phosphate onto it for NADP. Oops, NADP over here, which then to either one of these we can add a hydrogen, so we could get NADH. We know how that happens, during glycolysis or during the Krebs cycle. That'll happen, and we know that this, adding an H to this, NADPH, that happens in the pentose phosphate pathway. So we're not worried about those parts anymore. We're just looking at how we make these from tryptophan. Okay, so we're going to begin the process, right? So we need, remember, IDO, TDO, and KAT as our rate-limiting enzymes. Here is the kind of the this word, kynurenine. Kynurenine? Yeah, it's not that bad. I don't know why I struggle so much with this word, but it really does tangle my tongue. So here we have, this pathway. We require iron, so iron is necessary as a cofactor to activate some of our enzymes. And we're just going to, just take this entire pathway. We're adding in ATP, so we're using energy. At this stage, also at this stage. Here we have various enzymes, a bunch of different enzymes here. I have a fruit fly flying around here if you see me, flapping my hand around. There's a fruit fly. Here is where we have our PLP. So this is where we have transamination. So this is important that we have vitamin B6 for this particular phase. So just a bunch of steps, and again, you don't have to memorize the steps, but what you're looking at is that there is a lot of different stages to this process. We have a lot of different cofactors going on here, a bunch of different enzymes, not just the rate-limiting enzymes, but also other enzymes at each stage of this process, and we're using ATP for this process. And again, actually here. So when we're adding the P at the very end, if we're not satisfied with NAD plus and we're trying to get NAD plus, we'll use another ATP. So we're just adding a phosphate to our NADH. And there's another enzyme right there. So it's a pretty involved process, but important products here necessary for energy production and the electron transport chain. And this is important for lipid production. You can't start fatty acid synthesis without NADP. Okay. Okay, so here this slide is just talking about, remember what you use NAD plus and NADH for in the electron transport chain. And then remember what you use NADHP for, which is making lipids. It's also important for making reduced glutathione, which is one of those endogenous antioxidants. Now another place that you use NAD plus, which I haven't mentioned yet, is some second messenger systems that use ADP. So they're reliant on ADP. Also use NAD plus. So it's, to create, remember in second messenger systems you have just a cascade of chemical reactions, and NAD plus can be associated with some of those second messenger systems. So NAD plus and NADPH all have, a lot of functions. So some people will, take supplements of NADH to help create energy theoretically. Great. Now other examples of transamination. So remember, we're just looking at our PLP going into PMP. So this is sort of just other examples, things that we've actually seen before, but without actually looking at PLP. So there's PLP right there, and in this process right there. So that's our PLP, and remember, when it steals the amino group, it'll become PMP, and that goes backwards, right? So it's just taking the amino group from one thing and putting it onto another. So you can see that happening here between alanine, pyruvic acid, glutamate, alpha-ketoglutarate. So alpha-ketoglutarate would be losing the amino acid and giving it to glutamate. So PLP is stealing that. Same thing over here, same situation, alpha-ketoglutarate losing the amino acid, there's PLP. Here we're dealing with oxaloacetate and aspartate going the other way. So oxaloacetate loses the amino acid and we get aspartate. So just other examples of transamination, just showing the diversity of what PLP does and that we've actually seen it already. We've seen all of these processes already. We just haven't specifically mentioned PLP. Okay, now back to the rate-limiting enzymes. So remember the rate-limiting enzymes are TDO, IDO, and KAT. Here they are in the process. And actually, here we have our tryptophan. I know you can't see this so good, so I'm just going to explain it. Here we have our starting molecule, right? That's our tryptophan. Over here, this is the serotonin / melatonin pathway. So this is the other choice. We haven't explored this choice yet for serotonin to take, but we will in a minute. So we need, for this pathway to work, we need tryptophan dehydrogenases. Now, for the rest of it, so over here, this is where we need our TDO, IDO, and KAT. So since they're rate limiting, this means if you have a lot of them, you will do a lot of creating eventually NAD plus at the end, right? Which will then go into NADH and NADP and on NADP. Let's put an H on that to balance it out. So you can see here, there's TDO, there's IDO, and there's KAT. So TDO and IDO are right at the beginning of the process, right? So right as you go from tryptophan into kynurenine, I did it that time. So there's kynurenine. They're right there at the beginning of that process. And then there will be some intermediaries as you go through until you finally get to NAD+. KAT is on the side branch over here where you get a couple of different products going into this final end product. Now this is where the pathways sort of link up here, which is that this is a keto acid with varying uses inside the cell. But you'll notice that over here, this is the breakdown product, I'm running out of room, breakdown product of serotonin and melatonin. So if you can see here, MAO, monoamine oxidase, this is an enzyme that breaks down serotonin, melatonin, also dopamine, though dopamine doesn't have to do as our tryptophan pathway. It's handy to note here because monoamine oxidase probably sounds familiar to you from a group of drugs called monoamine oxidase inhibitors, or MAOIs. If you think about what an MAOI does, MAOI, it inhibits MAOs. MAOs, these things, break down your serotonin. That's your happy. So MAOs are the enemy of happiness in a way. Obviously, you don't want to have too much serotonin. That can cause serotonin syndrome, which can actually be deadly. So you don't want to have too much serotonin. So you do need these. These are not the bad guy. But if you have too much of this, you can end up with low levels of serotonin and depression. Melatonin, obviously, you don't want too much either because then you won't wake up. You'll just be very, very sleepy all the time. So you want melatonin at the right time, and you need to be able to break it down. So, these are handy so you can actually wake up. But if you have too much of these, again, you're going to end up having insomnia. Dopamine is another calming neurotransmitter, another kind of happy neurotransmitter. So again, you don't want to break it down too much. So for people who have, depression, anxiety, MAOIs treat depression and anxiety. Okay, so before we move on to the next slide, I just want to point out this product here. Before we get to NAD+, we have something called quinolinic acid. This is just an intermediary product on your way to NAD+, but notice there's an enzyme, like there's an enzyme for every transformation that a molecule takes. So that's what we're going to be talking about in the next slide, is this particular intermediary product. Okay, so what quinolinic acid does is it helps to stimulate your nervous system. You just don't want too much of it because it becomes a neurotoxin. It starts over-exciting the nervous system. So, you want the right amount of nervous excitement, but not too much. So let's say if this enzyme was malfunctioning and you just build up quinolinic acid, you're going to get what we call neurotoxicity. So that's just anything that is toxic to the nervous system, but in this case it would be overstimulation of the nervous system. And also you wouldn't have NAD+, right? So you would be kind of missing this. So you want to have this enzyme. You don't want to have too much of this because you want the end products, but you also just don't want to get overstimulated in your nervous system. Now, quinolinic acid, this is also an intermediary. Let's see over here, right? So from the KAT pathway, this is something that acts as an antagonist to neuroexcitement. So this is going to help slow your brain. Now, too much slowing is problematic and can cause psychotic issues. By psychotic, psychosis is an actual clinical term, so it's not just, not the way people kind of use it to just throw out like, oh, I feel psychotic, like you're like, but what it means is that you are having delusions that seem very real, hallucinations, just a real loss of reality. So those symptoms can be transitory, so you can have them at some times and not other times. They can be very disturbing. They usually would be disturbing because it's a loss of reality, so that would cause a person a great deal of distress. So having too much of that building up would dampen your nervous system to the point where it wasn't quite firing on all cylinders anymore and causing, possibly a psychotic break even, which is what it's called if someone has psychotic symptoms that become so severe that they just can't function through those symptoms. Now, remember that we need the end product of our tryptophan also for NAD, NADH. There's also, in addition to just our NAD, NADH, and NADPH, let's put that in there, your skin health is very, very reliant upon niacin, so vitamin B3. Your brain function, obviously, for all these reasons, is dependent on vitamin B3. Fetal development, as a person is being created, vitamin B3, among many other vitamins, is obviously really important. Now, having not enough niacin then would cause you to have skin problems, so dry, itchy skin, rashes, inability to heal, nervous malfunction, either overstimulated, understimulated, not able to develop properly if you're a fetus, so like not going through the normal stages of development or not developing fully. Now, you can have too much, though, and that can make you feel dizzy, constipated, have tachycardia, which means that your heart rate is too fast, which is uncomfortable. This, obviously, would be either one, really, is an easy fix. If you have too much vitamin B, stop taking so much vitamin B, right? Like, that's usually why. It wouldn't be dietary, typically. It would be somebody taking a great amount of vitamin B supplements because they are water soluble. So getting too many, your body can typically flush a lot of that out. But if you took way too much vitamin B, then that could cause, some of these symptoms. Not having enough means you need to eat more or ingest more vitamin B. Now, there are conditions where you, that are described by being related to a niacin deficiency. So pellagra dermatitis, this is a really unpleasant skin condition, full body rash, itching. We've seen this before in other situations. It can cause damage to your brain, not the skin problem spreading to the brain, but the pellagra condition itself can also cause problems in the brain. Hartnup disease, this is a genetic disorder. And here you don't have the ability to absorb tryptophan enough. So it's not a complete inability to absorb tryptophan, but it's that you're not able to absorb it quite enough. And so without the tryptophan, you're unable to make niacin properly. You end up having a niacin deficiency, and you end up with a similar symptoms to pellagra dermatitis. Okay, now let's get our tryptophan and turn it into serotonin and melatonin instead. So remember, we need our tryptophan dehydrogenases. So that's not on the slide, but we need to have those. And then we will run it through a series of hydroxylase enzyme, decarboxylase enzyme, an acetyltransferase enzyme, and a methyltransferase enzyme to finally get melatonin. Now the steps here, 5-HTP, that might sound familiar to you because this is a supplement that a lot of people take because it is a precursor to serotonin and melatonin. So the idea is if you take 5-HTP, you'll be more able to make melatonin and you will sleep better. And serotonin just makes you happy. So as supplements go, possibly it depends on how well you can absorb and how well your body will then utilize that and put it down this path. You have to have the proper enzymes for this. But the take-home message from this slide is that, 5-HTP, this is a familiar intermediary product because it's been touted so much as a supplement. You're taking tryptophan and getting serotonin, right? So that's nice. And then you're eventually proceeding on to melatonin, and I think everybody knows the link between tryptophan and melatonin. Everybody's like, oh, I got so much tryptophan and now I'm sleeping. Now, the fun part of this, or the funny part of this, is that's actually not quite accurate. People, get sleepy after Thanksgiving dinner, and they think like, okay, I ate the turkey, I got a lot of tryptophan, and now I'm going to sleep. But it doesn't work that fast. Grandpa is passing out well before he's digested the turkey and absorbed the tryptophan, much less got it to the cells and started the whole tryptophan to serotonin pathway, or tryptophan to melatonin. So what's actually happening there? While the tryptophan will help Grandpa make melatonin after some hours, why Grandpa is falling asleep a couple hours after Thanksgiving dinner is because like when you're really stuffed, a lot of the blood is going to your stomach and not your brain. So really, you eat a lot of anything and you're going to get sleepy. Just as a fun word to throw out there, postprandial narcolepsy. This is falling asleep uncontrollably after eating. And it is due simply to where the blood is. The blood, when you are super full, the blood is going to your stomach and you don't really have much left for your brain, so you feel sleepy. You'll probably notice anytime you eat after lunch, a lot of people get in a slump. You know, whole countries have entire cultures set up around a siesta where you eat lunch and take a little nap. It's because of that. Okay, so remember we were talking about our quinolinic acid as a neurotoxin. I just want to revisit that really quick. NDMA, or another one that trips me up, NMDA receptors are in the brain. They are excitatory. Remember we said that quinolinic acid was a excitatory sort of thing that is toxic in too high of concentrations. So these NMDA receptors are excitatory, and quinolinic acid is an agonist for these, which means that it helps to bind to them, or it can bind to them and stimulate them, to stimulate the brain. Now, these NMDA receptors are actually glutamate receptors. So glutamate is a neurotransmitter that is stimulatory by binding to these. So we can have quinolinic acid binding to the NMDA receptor and stimulating it. We have glutamate binding to the NMDA receptors and stimulating it. Now, another thing that quinolinic acid can do, in addition to just stimulating those glutamate receptors here, these NMDA receptors, it increases how much glutamate is released. It stops the glutamate from being taken back up by the neuron, so it leaves it in the synapse. So it's just increasing not only what it's doing itself to the NMDA receptors, but also just the glutamate and what that is doing to that receptor. So together they are helping to stimulate the brain, which is great. But if you have too much, that's neurotoxicity. So that can lead to degeneration of your neurons. It can lead to convulsions if it's really severe, strokes, epilepsy, so seizures, which kind of close to convulsions. So there are certain drugs that actually hit those receptors, like ecstasy. So that is a neurostimulatory kind of drug. It makes the brain fire in ways that maybe aren't quite associated with reality. So, it's a recreational drug because as long as you don't take too much, you don't want the neurotoxicity of that. Just, remember, someone taking ecstasy is stimulating these receptors in a way that they're not meant to be stimulated, and it can cause neurotoxicity. Now, there is another drug called ketamine. Ketamine inhibits those receptors. So it dampens activity. Obviously, too much is going to slow you down to a point where, that's not healthy. This is used in veterinary science. It's a horse tranquilizer. Horses weigh 1,000 pounds pretty much, give or take some. So ketamine is very powerful as a tranquilizer. But ketamine has gotten some interest lately in mental health research in terms of microdosing. So that's small amounts of hallucinogens to treat depression, anxiety. So is ketamine a hallucinogen? A little bit. It's inhibiting the nervous system to the point where, it's slowing down so much that if a person were to take the so-called right amount, then there could be some mild hallucinating. But apparently it's shown some promise for actually treating depression and anxiety just by slowing down the nervous system. Particularly with anxiety. Anxiety is a overstimulated nervous system. The nervous system is working overtime. So ketamine in small doses, very small doses. Remember, this is used to knock out a horse. So very, very, very small doses have some promise. It's not, FDA approved yet, and it's not been fully clinically studied. So I'm definitely not recommending trying that until more research is done. It's just an interesting avenue of research. Okay, and just to finish off the lecture, these are people who have serotonin, right? They're happy. So serotonin is the happy hormone. Okay, so that's it for this lecture. Our next lecture will be the ornithine cycle for urea that is, related to amino acids as well. But that's it. I'll see you next time.